All right, it's my great pleasure to introduce Simon Verleen today, who's today's speaker. Simon's been very good at updating us about his progress over the years and months, so I won't spend a lot of time on his introduction. Just say he's a native of France. He's got his bachelor's degree in 2010 from the Pierre Marie Curie University in Paris, and then moved to Montpellier to complete his master's degree from the University of Montpellier with IRD in 2012. And then after that, he worked as an orchid specialist for the Missouri Botanical Garden based at the Muséum National d'Histoire Naturelle in Paris. And since 2018, he's been a PhD student here at the joint CUNY-NYBG graduate program in plant science. And today he'll be presenting a summary of his doctoral dissertation work. So warm welcome to Simon.
Thank you, Greg, for the introduction. Beforehand, I'd like to acknowledge my committee for helping me greatly with this work and making it what it is today. So I'll present the results from my doctoral dissertation titled The Last Stand of Angræcum, Biogeographic and Evolutionary History of Darwin's Muse. This work is the continuation of my previous endeavors with orchids in Madagascar and continental Africa. Oops. Excuse me. So I obtained, as Greg said, I obtained my bachelor's in biology at the Pierre Marie Curie University in Paris, and then a master's degree in tropical botany at the Muséum National d'Histoire Naturelle, also in Paris. And for my master's, my research project was focused on Bolusiella. So as you can see here, Bolusiella are small twig epiphytes from continental Africa with very small flowers, with Vincent Droissard from the IRD and who was based at the time in Yaoundé. So Vincent introduced me to fieldwork in Cameroon and the shade houses. But what is a shade house? The shade house is a simple answer to a major problem that impeded orchid systematics. In Central Africa, it's estimated that 70% of epiphytes are orchids, and at any given time when you're on the field, more than 90% of these are non-flowering. And so that impedes you from identifying this plant to its species. And on top of that, historical collections are oftentimes poorly preserved, with flowers missing, broken, and the pressing usually obscures diagnostic characters that would help you identify these specimens to its species, and also precludes the description of new species. So what if you could preserve even the smallest flower with its 3D structure and diagnostic characters, as you can see maybe down there? Well, that's the point of the shade houses. Think of them as the tropical equivalent of a glass house. So plants are brought non-flowering from the field in the shade house. They're then cultivated on slabs of wood as they would grow as epiphytes in their natural environment. When they are cultivated, daily watered, and when they flower, then we can collect pickled specimens of the inflorescence, a little bit of leaf. We can also do silica dried leaf samples for future DNA extractions, and pictures to have a record of color, for example. So not only the shade houses are a great tool for these systematics endeavor, but also for long-term ex situ conservation. Because you have all these plants readily available from within a single species, you can then cross-pollinate different individuals and then have them produce fruits from which you can collect the seeds and store them into a seed bank. On top of that, fantastic training ground for the next generation of orchid specialists, the same way I was. The shade house cultivation technique was perfected in Gabon, and then was expanded into different countries in continental Africa and Madagascar by the Missouri Botanical Garden and its partners. So there are currently 9 active shade houses in 5 African countries and Madagascar. It's estimated that more than 30,000 living plants have been put into cultivation in these shade houses, and they yielded 24,000 specimens that have been identified to over more than 500 species. That's about 20% of the orchid diversity in continental Africa and Madagascar. In Madagascar alone, the shade house network started in 2011. And as you can see here on the graph, so it shows the number of specimens collected. As you can see, there's a clear growth in 2010, 2020 for the orchid specimens collected on the island. And as a result, only in the past 4 years, 13 species and 2 genera have been described based on specimens that were collected in these shade houses. But let's take a step back. I want to give you a little bit of a refresher on orchids, and then we'll talk more about the group I've been studying. So vegetatively, the majority of orchids are sympodial. That means that they grow a new pseudobulb every year. But angraecoid orchids, the ones I studied, are monopodial. That means that they only have a terminal meristem. The flower, orchidologists like their peculiar names. So the flower has a first whorl of 3 sepals, a second whorl of 3 petals. One of them is modified and called the labellum. And then the reproductive structures per se are contained within the column that is here in the middle. And you can see over there on top, the pollen are aggregated in pollinium. And then the pollinia is attached to a visidium, which is a sticky pad that will stick to the insect when they visit. And the entercap comes on top and protects all of that. On top of that, angraecoid orchids also have the specific appendage on their labellum, which is a nectary tube. We call it a spur. The spurs can be either saccate or very long, tubular usually, and they have nectar at the bottom. So flower of orchids can be non-resupinate. That means that the ovary and the pedicel does not change its orientation, and then the labellum here is top facing. The flowers can also be resupinate. That means that the pedicel and the ovary will undergo a 180-degree torsion before the flower opens and puts the labellum into a lowermost position. And in angraecoid, you then have again another torsion of 180 degrees that makes a complete rotation and brings the labellum uppermost again. And so the ovary and the pedicel are really completely turned, twisted. Thank you. So why would you have these different lowermost, uppermost orientation of your labellum in a single group? Well, it can be explained when the pollinator comes around, and if the flower is resupinate, the pollinia will be stuck on its dorsal side, and if the flower is twice resupinate, it will be, or even just non-resupinate, will stick on its lower side. So that prevents hybridization between 2 species that would have the same length of spur or the same shape of a labellum, but it also promotes diversification, as then you exploit more space on this insect, as you can see here on this sphinx moth. So sphinx moth, white flowers with spur, that might ring a bell. So within Orchidaceae, which is one of the largest flowering family of flowering plants with 31,000 species, 1 out of 10 species of angiosperm is an orchid. Darwin and Wallace Darwin at least received flowers from James Bateman from Angraecum sesquipedale. And when he saw these flowers with this long nectar spur of around 30 centimeters, he hypothesized that there must be an insect that has a proboscis that would match this length of the spur so that it could access the nectar. A few years later, 5 years later, Alfred Wallace expanded on it and really tried to explain the mechanics that would make this happen. Here, I love this illustration on the left that was done in 1867, the same year Alfred Wallace expanded on this prediction. And as you can see, the artist just drew whatever he thought the Sphinx would look like. And finally, the Sphinx moth was indeed discovered in Madagascar in 1903. But the pollination itself was not observed until 1975. And so this illustration on the right was done in 1973. So again, still the pollination had not been observed at that time. Here is the distribution map of Angræcum. They're mostly in Madagascar, but also in the archipelago surrounding the island. So the Mascarenes, the Comoros between Madagascar and Africa, the Seychelles. They're also in continental Africa, of course, the island of the Gulf of Guinea, and two species, one in Socotra and one in Sri Lanka. Being distributed in Madagascar, mostly in Madagascar, it means that they are facing heavy threats. In Madagascar, we estimate that the forest cover has reduced from, in the last 50 years, over 40% here in green. And this reduction of the destruction of the habitat is in part due because of slash and burn agriculture. So when you cultivate in a forest that you've cut and burned, you have access to a lot of nutrients for the rice, for example. But then after a few years, the rain washes it all off and you have to start again. Charcoal production for cooking, but also for heating at a very local level. But on a broader scale, you have major impacts like mining, but also precious wood exploitation for ebony and rosewood, for example. And because we're talking about orchids, the list wouldn't be complete without talking about poaching. So previously this year, we published a very rare species, Solenorchis impredicta, the first Solenorchis in Madagascar. And 3 days later, it was on a page on Facebook, the page of a collector. This species is only known from 2 localities that are heavily protected. God knows how they got it. If we look at the evolutionary context of Angræcoides, here is the tree of land plants. And within the monocots, the order Asparagales, this is where we are. And finally, within the orchids themselves, we're in the subfamily Epidendroideae, the most diverse subfamily with 25,000 species out of 31. So it's an enormous group. And these are the most, some of the most recent diverged group in the orchids. Concerning Angræcoides themselves, since 2006, they've been studied, the phylogenies have been published. I've synthesized the results here. So Angræcoides is made out of 3 main clades. The Afro-neotropical Angræcoides, as the name implies, are mostly distributed in Africa and the neotropics. Sister to Vici-Angræcoides, a group from Madagascar and the surrounding archipelagos. Both of them sister to Eungræcoides, also from Madagascar and the surrounding archipelagos. So here we find these 3 groups once again, but one of the problem, of course, is Angræcum. One of the most iconic and most diverse group is not only polyphyletic, but also split between the Eungræcoides and the Vici-Angræcoides. So some species are very distantly related. Regarding its biogeographical history, previous authors have worked on either a subgroup of species or a specific region. So Michneau and collaborators in 2008 worked on orchids from the Mascarene. Pessoa and collaborators in 2018 worked on the neotropical component of this group. And finally, Dayer and collaborators in 2021 on the species that occur in the Gulf of Guinea Island. However, an hypothesis for the center of origin of Angræcoides as a whole has yet to be tested. So if we summarize a little bit what I've said already is Angraecoid orchid center of origin has never been tested. A natural classification for its most iconic and most diverse group has yet to be proposed. And finally, a group of Angraecum species distantly related really needs to be investigated. Hence, the three main chapters of my work split in three different hypotheses, and we are going to focus on the first one, that I hypothesize that the center of origin of Angraecinae is Madagascar, and the specific objective to do so was to reconstruct a phylogenetic relationship within the group, estimate time divergence, and finally reconstruct ancestral area. A little bit of methodology. I pooled together a lot of sequences from 6 loci, 5 plastid, and 1 nuclear that were already published in those previous studies that I mentioned before. But I added 46 new individuals out of the shy of 1,000, 38 new taxa out of the 500, and 7 new genera out of the 45 that makes this most complete data set to date. And all these additions were mostly orchids from Madagascar. Concerning the phylogenetic reconstruction itself, we used a Bayesian inference and maximum likelihood frames. The time divergence estimate was done with BEAST and with 2 secondary calibration point. I'll touch on that later. And finally, the ancestral area inferences were recovered with BioGeoBears. So the first results showed that indeed we were able to recover this 3 main clade structure of the Angraecoids. Not only that, but also that Angraecum was indeed polyphyletic, and that 3 small or small monotypic or small genera from Madagascar were nested within this big Angraecum clade in which the type species falls. So that would be Angraecum sensu-stricto. And finally, a little intersectional relationship within Angraecum was recovered, precluding us from making more evolutionary hypothesis on the history of this genus. Divergence time estimate. So as I said previously, we used 2 secondary calibration points. The problem is that the first relative with in orchids with a fossil is very far removed in the tree, and so I would have had to add a lot of outgroups, more outgroups that there are in groups, and so at some point it would have muddied completely the analysis. So I chose the first secondary calibration point here at 24 million years ago, which is the split between Angraecinae, the Angraecoids, and its sister clade. And the second one was the emergence of the island of Réunion 2 million years ago. As you can see, as a result, we found that the crown age of Angraecinae was around 21 million years ago, which makes it a mostly recent group. And for the 3 main clades here in green, there are middle Miocene, their crown age is middle Miocene, and major speciation rounds happened in the late Miocene and Pliocene, Pleistocene, the last 5 million years. And that's in line with findings for not only other groups of plants, but also with the geographic events and climatic events that happened in Africa, where during those past 5 million years, there's been a bunch of pluvial and displuvial events that have contracted and expanded the forest in sequence. Reconstructing the ancestral areas. Here's the result from BioGeoBears. It's a little bit crammed, I'm sorry, but I've synthesized it here, and we can follow up with a map. And what you can see here is in number 1, the node for the Angraecoids was recovered as being in Madagascar, so the center of origin in Madagascar. And then in 2, the colonization event from Madagascar to continental Africa that explains the whole diversity for the Afro-Neotropical group. And subsequently, another colonization event from continental Africa towards the neotropics that explains all the neotropical representative of this group. Several independent colonization events from continental Africa towards the three main archipelagos surrounding it. So the Mascarenes, Seychelles, and the Comoros, and one in Sri Lanka. And finally, three independent retro-colonization events from continental Africa to Madagascar. One at least have been followed by another round of diversification. So now that we've seen the overall picture of evolutionary and biogeographic history of Angræcode orchids, let's focus a little bit more on Angræcum. So that was the point of my second chapter. As I shown before, Angræcum was shown polyphyletic constantly, but there's still a group that can be recircumscribed. And so to do this and to recover sectional limits and intersectional relationships, Sanger sequences didn't cut it. So I intended to produce a comprehensive next generation sequencing data set with target sequence capture, and more specifically the Angiosperm 353 probe kit. So I chose this one specifically because the PAFTOL project already have sequenced a bunch of orchids with Angiosperm 353, and also it was cheaper than the orchid specific one. So, and with that, with the phylogenetic inferences that arise from this data set, I would propose a natural classification for the genus. Concerning sampling effort, a little bit more than 300 individuals were sampled at the Brussels dried leaf collection, and or also in the living collections in Madagascar. So I would run around the shade houses, collect samples and vouchers, and so I could use them here. I also took 14 individuals from herbarium sheets in Paris that were collected in the 70s, so not too long ago. And I also added 24 individuals that were already sequenced and present in GenBank from the PAFTOL project I mentioned before. Concerning Angræcum, I managed to sample all 16 sections, and it represented around 96 species, which is about half, a little bit more than half of the genus's diversity. Extractions, 334, so mostly with CTAB, a little bit with an acetone protocol that did not work for me, at least. 192 samples were sent, and out of the data that came back, I was able to assemble 152 individuals. Concerning recovery, so here on this heat map, let me walk you through it a little bit. Every line is an individual, and every column is loci. So you have 353 loci, and you have those 152 samples. So when you have a white line, that means that the sample just didn't work. And when you have a white column, that means a loci didn't work. Out of the 353, we were able to recover sequences for 350 loci, which I was very happy about. And if you see here, those lines that are a little bit lighter, these are the specimens from that were on GenBank from PAFTOL. So I was also happy to gather a little bit more data than was already there. Concerning paralogs, so in such a recent group with such an enormous diversity, you would expect that what would have happened is either hybridization or gene duplication events. And what that would happen is that you would have a loci that would duplicate, one of them still being tied to its function, and so would be tied to its function. So would have to stay the same, whereas the other one is not tied to the function and could have a different evolutionary history. So what you want is to make sure that those single copies or at least little copies you have are from the gene itself and not from a duplicate loci that has a completely different evolutionary history. And as you can see here, the lighter the better, and still each individual is a line, loci is a column. A few loci had a little bit of paralogs, but I didn't complain. So the first phylogenetic inference I did was to concatenate all these loci together and to run it in IQ-tree with a maximum likelihood algorithm. And what the first result was that the structure in 3 main clades was respected. So again, I was pretty happy about that with the Afro-Neotropical Angraecoids, the Vitzi Angraecoids, and the U Angraecoids. And then I started to flag Angraecum species, and I didn't only have 2 clades, now I had 4. So I had to try to figure out what was going wrong. So clade 1, it's a very cool example. I had 3 samples of what I thought was Angraecum rancoglossum, and in the tree, 2 of them fell far away from other Angraecum species, and 1 was right smack inside. And so I figured either those, it was an error, I would have mislabeled them, I would have mixed up my pipetting or whatever. Always me the problem. But when I checked all this, recovery was good. And so then I went back to the vouchers. That's why vouchering is so important. I was able to check the plants, check the pictures, check the flowers. And indeed, vegetatively, they had a few differences, but on the floral morphology, it's very hard to see those differences. There are a few characters in the petals and the sepals, but it's very minute. So this is a great example of a convergence of shape, floral shape, and probably driven by the pollinator. So because they're trying to attract the same pollinator, you need to do the same thing. And at the end of the day, this is what you get. And this is also important in terms of conservation, because I'm walking around, I'm seeing a flower like this, I'm like, oh, this is Angraecum rancoglossum. It's grass, it's everywhere. So I'm not collecting that. Meanwhile, what I'm missing is probably Angraecum pergracile, which is it's a rare species. The type has been destroyed in the Berlin fire. There's around maybe 3 points in Madagascar. So thus, this is how the phylogeny can work back to the taxonomy, and then both of them inform conservation. Then group 2, I'm not going to mention it right now. It's those clade of distantly related species we had with Sanger. I was fine with that. Now 3 and 4, I was a little bit more concerned. So in 3, you had Angraecum species that were sister clade to Jumelia species, another group of Angraecoids from Madagascar. And then in D, what you could call Angraecum sensu stricto with the type species. Looking at this clade 3, sorry, it was very morphologically, it made no sense. Jumelia has a very clear floral structure with a clear insertion between the labellum and the column that those species of Angraecum did not have. So already I had to figure this out. And then, as I said before, 4 would have been Angraecum sensu stricto because it has the type species. So because I concatenated all these loci, again, each of these loci probably have different evolutionary history. And so when you concatenate these all together and you run them to produce a tree, it will infer that all of these have the same evolutionary history, and thus maybe the modeling we had before. So I used coalescent-based a coalescent-based program, Astral, to do a tree for each of these different loci, and then have a summary tree of all these different unique trees. And in this framework, what I had was more in line with morphological data. So clade 1, Angraecum rancoglossum, okay, we made our bed. Clade 2, still there, that's fine. And here we have those Angraecum species that were with Jumelia, now back with those other Angraecum species. And this was in line with what we had with Sanger, what was published before, and morphologically, it made sense. If we zoom in on 3 and 4, which is Angraecum sensu stricto now, concerning those small monotypic or small genera that I mentioned before, we verified that 2 of them, Sobenicophia and Eonyila, indeed nested within Angraecum. The third one, Lemurorchis madagascarensis, was only distantly related. And that's not what was found previously with Sanger sequences. Now, the 2 problems with that. The first one is all these Sanger studies used one individual. The voucher, I couldn't see it. I don't even know if it exists. My voucher, it's a plant that was not flowering. So at the end of the day, I'm not sure if Lemurorchis is indeed or not in Angraecum, but based on the best evidence I have today, it is not. And so I'll discard it until I can add more individual from the species. Now, if we look at Angraecum's sections, what we see is that most sections are polyphyletic or paraphyletic with one another. So for example, in A, that's a group of species mostly from section Pectinaia, but within it, you can find species for Phyllanges pseudojumelea, same with 6 and C, et cetera, But when you look at these group of species and you look at their characters, those clades make sense. So for example, let's take clade B and F, for example. So this was one section that is now split in 2. As you can see here on the pictures, clade B has its labellum lowermost, so the flower is resupinate, while the species in clade F have their labellum uppermost, so they are twice resupinate. And so I look at those species that were previously put in the same section. Now I see them separated, but it makes sense. Here in D, for example, you have section Amblotia with species from Perrier angraecum. But when you look at these flowers, the entrance of the spur is in a funnel shape, and this is not present in other of these groups. So for each of these sections, I was able to, you could find diagnostic characters that would make them a section based on monophily, but also these characters. And then group G, so this is a mix of 8 different sections. They all have greenish to yellow flowers, very peculiar. This is 1/3 of the Angraecum diversity. So you think of Angraecum as big white flowers with long tubes, and meanwhile, 33% of these are just smaller plants. And I was not able to find within this group diagnostic characters that would help me to go at a finer level. So I lumped everything in one section. And what probably happens is that here you have a pollinator shift from moth or bigger insects to small, for example, midges, but we don't know because we never characterize the pollinators for these plants. And so because of this new group of insect, you can then exploit a new set of niche and then radiate like that. If we look, so this is the result of the systematic treatment I chose to apply to Angraecum. So overall, 188 taxa, 179 species. They are distributed in 11 sections out of the 16 previously published. And we took out 13 species and brought 5 species in, notably from the previous genus Sobenicophia, who's now a section, and Eonyila, same thing, is now a section. And finally, the third hypothesis, my third chapter, this group of divergent species I shown in Sanger, both in Sanger and next generation sequencing that were diverging, represent a new genus. And so for that, I went to identify synapomorphy for this group of species and describe it as a new genus, Vavavoluin. Rolls on the tongue. So here it is, clade 2 previously, those small plants, small flowers. One great example of it is Angraecum tenellum. These small white flowers, very minute plants. greenish yellowish center. The only problem with those synapomorphies is that they're everywhere in the group, not only in Angraecum, but also in the outgroups. Campylocentrum is in the neotropics. Microcelia is also in Madagascar. So again, I had to take my finer tools and dissect the flowers. What we did, I had previously worked on that group. So we described 2 new species in 2016. In 2019, during a fieldwork in the Eastern Escarpment Forest, we stumbled upon something that looks related, but I couldn't identify to a described species. And finally, in 2020, we described Angraecum polyphemus, another kind of related species. So from the plants from the individuals that fell into that specific clade, and with all these specimens that I saw before, I tried to look for synapomorphies. And I did find a few, 4 to be precise. The first one is the trilobe labellum. Even obscurely so, oops, that's not the correct image. Anyway, they have a labellum that is trilobed and that has hairs at the entrance of the spur. The rostellum is deeply cleft with acute lobes, and the anther cap that protects the pollinia, has kind of a beak. I considered around 20 species that were in 5 different sections of Angraecum, and I went along and dissected all these flowers. And what I found was that out of these 20, 14 taxa are indeed, share indeed those synapomorphies. 9 described species, the taxonomic novelty we found on the field. 4 additional morphotaxa for which I don't have enough material, only 1 flower, or even just a picture from a collector. And so I couldn't do much with them. And 5 heterotypic synonyms fall under Vavavoluine tenella. Concerning their distribution, they're mostly distributed in Madagascar, but also in the Eastern Arc range in Eastern Africa, all the way to northern South Africa, and 1 species in Réunion. This species in Réunion is also present in Madagascar and in continental Africa, and it's Vavavoluine tenella. So if we circle back to our hypothesis, the center of origin of Angraecinae is indeed Madagascar. We updated the phylogeny for the group. The divergence times were confirmed based on what was previously published. We were able to recognize a monophyletic Angraecum genus. Angiosperm 353 proved very valuable in getting the breadth of character we needed to recover intersectional relationships too. So then now we can work more on evolution within Angraecum. And finally, so we redelimited the genus and its sections. And finally, this group of divergent species was indeed a new genus. We found some strong synapomorphies for it and described it as Vavavoluine, new genus for 10 species in the Vitziangraecoid. And Vavavoluine means hairy mouth because of the hairs it has at the entrance of the spur in Malagasy. What would be the next step? So this clade, the Vitziangraecoid, where the Vavavoluine fell, but also Angraecum rhynchoglossum, and a few genera that are polyphyletic with one another would be very interesting to revise and to check all these different species. I also like to characterize pollination syndromes in Vavavoluina, those full small white flowers that seem to have convergent clades in all over the group, and also Angraecum section Lepervenchea, those tiny Angraecums with yellow and greenish flowers that exploded recently. And finally, do some population genetics. It has been done with Angiosperm 353, and I would like to characterize what structures the population of those small species that have probably small pollinators. Is it we don't expect those pollinators to travel long distance, but the problem with and orchid seeds are known to travel long distance, but they're still tied to their mycoheterotrophic partner. And so what would explain such a big distribution of one species, Réunion, Madagascar, and South Africa, maybe the pollinator could surprise us. I don't know. And I tried to do this. So for the pollination, I tried to do this during my PhD. Didn't end up too much, but just this cool gift. Wow. Finally, I'd like to express my my thanks for my group of colleagues and friends that work all over in Madagascar, in continental Africa, in Brussels, Montpellier, here in New York. And this work would have not been possible with what they did before. And hopefully we'll do more together. And I'd like to thank the institution that have allowed me to visit, open their door, and allowed me to use their collections, and the financial partners that made a bunch of field work, but also travels possible. And a shout out to Léopold Dagalier, who helped me a lot with those NGS analysis. Brigitte Ramandi-Binsua, who's my mother in Madagascar. She takes care of me like nobody. And Jouan Farmignon for the great discussions we had concerning angraecoids. Thank you very much.
Thank you, Simon. We have plenty of time for questions. If anyone has.
Well, first of all, congratulations. Was very cool. So I was wondering, we were talking about the how the migration of the different angraecoid groups. And everything you said that most of these like speciations, events, and everything happened during the Miocene, right? So where the land masses were somewhat already apart. So how is that you think these events of movement
happen? So as I mentioned, orchid seeds are basically just a small mass of cells within a little bag. And so what happens is that when they're carried by the wind, they can travel long distance. And so those long distance dispersal events are for orchids, like they would be for ferns, are not really something out of the ordinary. However, as I said, it's not because they can travel far that when they will fall somewhere, they will have to germinate. And the problem with orchid seeds is that they cannot germinate by themselves. They need a mushroom to break the seed, this coat, and then help the seed germinate. They will also be in partnership. So the mushroom hypha will be in the orchid, etc. So there's a balance between they can go everywhere, but then they need their partner when they arrive somewhere. And so that is why probably you don't have angraecoids worldwide. Also, it could be explained by niche occupancy. So for example, if the niche is occupied by another group of plants somewhere else, it's difficult to settle because it's basically, it's already, your niche is already occupied. But as I mentioned, some of the small plants are understory obligates, and they seem to be able to traverse long distance. Whereas in an understory, you don't expect a lot of wind or a lot of those seeds to travel long distance. So that's why I want to do the thing with the pollinator. So yeah, I hope it answers your question. Yes.
Yeah, congrats on your work. It's really
Thank you.
great. And I really think that you have set yourself or set the framework for now testing the hypotheses that I think are super interesting, as linking pollination biology, population structure, and the spiritual affinity of the plants. Speaking of that, I was very interested when you mentioned the two species that are strikingly similar with yellow flowers, because they do fall into very separate places on the tree. So I was wondering if they are sympatric or are they overlapping distribution? Because if that were the case, I would have expected that because of gene flow and the similarity in the, well, similarity of flowers would have promoted gene flow, and then that would have caused in methods like IQ3 to cluster them together because we're not accounting for the gene flow there.
Yes, so you mean those two the Angraecum species and the Jumelia that were lumped together with the,
I think it was 200.
because those two Angraecums, so they're distantly related, and it makes sense in morphologically, and so they are symmetric. They do occur in the same area. So, for example, I found both of these in Ambatouvi. In the Eastern Escarpment Forest of Madagascar in that center. And so there must be another barrier, which is not morphologic, but that could be genetic, that prevents hybridization between those two. Because, well, there's only 3 individuals, so I don't know. But maybe if we added more individual, maybe there would be introgression between those two things, but I haven't seen it here. No. Yeah.
Really fantastic talk. As I'm sure you know, there's often a lot of discussion in work community about what is a genus, and we have big genera or little genera, and I really appreciate your discussion about that. I'm wondering, like, what was your sort of philosophy that you followed in terms of how you demarcate genera, particularly within Angraecum, where it seems like you have morphological synaptomorphies for each of these clades.
Yep.
There are sections, why sections, not genera. Yeah, where you just expand on that.
So, first, within Angraecum, I was a little bit more conservative, so I decided to keep everything as one genera, Angraecum. It has been split before by our colleague Darius Sachetko. The problem is that he split, the way he split it was based on an ITS tree. And so every single genus he created is polyphyletic. Mm-hmm. So the problem is you create this work for colleagues in herbariums to split all these genera. Meanwhile, all has to be lumped together because it's not good work. That's one problem. Whereas the sections are just a working tool for me and the other colleagues working on Angraecum. Everybody else can discard that. For the small genera, the Vavavouluine, why make it so it's a sister clade to Lemurella, another group of Angraecoids that do not share those synapomorphies, and that looks strikingly different. I don't even need to have the flower at this point. I can see the plants and I can say, oh, this is probably Vavavouluine, this one is probably Lemurella. I looked at all the other species in Vitziangraecoides, so the same major clade in which it is, and it's strikingly different. It is very small. The other ones are medium to large plants. Some of them might have a trilobe labellum, but not the hairs, and etc. So at the end of the day, because they were so morphologically different and created such a well-defined unit, then I decided that that's probably a genus and not something else. That was my philosophy. Yes.
Simon, I'm always so impressed by how much work you do. And about such diverse group. Can I ask a general question about, well, why do you think orchids are so diverse? I know pollination is part of the story here. And can you relate that a little bit to the shade house part of your presentation, which I thought was super interesting, particularly as a research tool, and you explained that really well. But I wonder if, given that these things can evolve so diversely and make so many different species, is there any concern that we might be making new species by putting these disparate things that wouldn't otherwise be together into a shade house for our purposes?
So the first part of the question is why orchids would be so diverse. I think one of the point, as you mentioned, is pollination biology. As I shown in Angraecum or Angraecoids in general, they can switch from one pollinator to the other by simply also just resupinating once more. And so what happens is that then you can exploit a new group of insects. And within this new group, you'll have larger, smaller, etc. And then you can have species that will look alike, but will be sufficiently different that it will exploit a genus or a specific species of pollinator. Then you have the great not the invention, but the use of epiphytism, which makes it a group that can either live on the ground or live in a drier environment. On the bark of a tree, it's very dry, and so then you can go to drier from this epiphytic lifestyle. You can go back to a drier environment on the ground, and so you can exploit different ecological niche from there. The fact that some have pseudobulbs, this is also a great adaptation to drought, and so you can go into semi-arid forests or grasslands, etc. What else can I think about?
I mean, many other groups could have those same characteristics too, right? Is there something about the way the genetics of orchids work that facilitates diversification?
So I think there's a potential for hybridization too. So in the wild, some groups will hybridize a lot. We can see that with terrestrial orchids, mostly in, I know the example of terrestrial orchids in France that frequently hybridize with one another. And so you create diversity like this by hybridizing, creating something new, will make a population, will be able to settle, etc. So that's one thing. And then gene duplication events. So they, I don't know exactly, but I know, for example, that in Angraecoids, there was a gene duplication event before, and then they just been reducing a bit those extra chromosomes. But if you have gene duplication, then you can create new functions, you can create, and then you can go to new insects. Yes, yeah, sorry. And so, yeah. And concerning the shade house, so the pollination are mostly done by hand. And so usually the people in charge of the pollination, they either know the species or they've been identified by at least two person. And so the data is online. They can look up, oh, I have this individual flowering, this individual, they look the same. Do they have the same name? If so, I can cross-pollinate. The presence of pollinators naturally within the shade house. So most of our shade house are outside the forests, or at least a few distance from the forest. So most of the pollinators most of the plants do not produce fruits naturally. Some of them might because of auto-pollination, because we don't know, but a bunch of them can do that too. So we try to take the maximum step to be sure. Also, every time a pollination is done, there's a tag, so we can trace the fruit. And so, for example, if one day we identify two plants were identified the same, and for example, the example I had with we realized that, oh no, they were not the same, then we can take out this specific seed set and not use it for conservation or rehabilitation. Yes.
Especially in the wild. So when you're exploring in the wild and you find these plants, how often do you find fruits on them?
Oh, it's frequent. It's not rare.
And do you think that's mostly because of self-pollination, or can't you tell?
There's no way to tell. The only way we could tell is certain groups for which we know there is self-pollination. So, for example, some Jumellia species have dispersed from Madagascar to Réunion with a long spur. But in Réunion Island, the diversity of huge sphinx moth is very reduced. And so Claire Michneau, which I mentioned for her biogeographic history, also looked at those Drumelia and cross-sectioned the flower and saw that all the organs that were made to prevent self-pollination were very reduced. So we would have to look flower species by species to see if those structures are indeed well developed, like I shown in the Vava voluine with the rostellum, with very deeply cleft. So this is usually not a sign that it autopollinates. The rostellum is usually very shriveled or very small. And then it helps the pollinia to be in contact with the stigmatic surface behind when the flower shrivels. What was I going to say? So we would have to look species to species, but then no, if you're on the field and you see a fruit set, usually we take it as a sign that there was cross-pollination. So when we're on the field, we also collect those fruit sets. So we put them in the seed bank. And so what happens is if the fruit is well mature enough, we'll take it. And usually if, for example, in long inflorescences, if pollination happened between two flowers in the same inflorescence, the fruit will not develop. And so this is also another barrier to prevent selfing in some species.
So I'm especially intrigued when you mentioned that you tried to do some observations in the field yourself. Were you able to do night observations?
Yes, so we have Vincent Droissard, my advisor for my master's. He developed a whole under $100 box, which is a camera trap that can film day and night. Those are linked to a battery pack, like I have for the phone. And so they can last for around 24, 48 hours. But he also linked them to solar panels. So now the only limiting factor is the SD card. And so basically they can film for hours on end. And so then we have day and night observation. And for angraecoids, we usually try to look more closely at the night time.
But what about the tiny flowers? He's even able to make observations with ambient light on these tiny flowers, or this is just larger flowers?
No. So for example, the GIF I had at the end, oops, sorry. There you go. This flower is maybe, I don't know, less than 5 millimeters long. And so with those tiny flowers, we're able to put the camera on it. I was able to see ants go running around, not scale insects, scale insects that would also run at the base of the leaves, etc. Unfortunately, no pollinator, but we can. There is enough resolution for us to characterize a pollinator if we ever
Even at night.
Even at night.
Okay,
Yeah. I don't have unfortunately, I don't have a nighttime video, but we could at least, maybe not the species, but at least the group. And that would be a start.
But no hits.
No hits. I didn't film, personally, I didn't film that long because we didn't have long to film. I filmed most of them in the shade houses too. So that's another limit because the shade house don't have all the same insect breath that the wild has. But hopefully one day I'll be able to bring them with me on the field and leave them there for.
I think the opportunity that you had to do a lot of the pollination work was in the middle of COVID and everything. So there's a lot of.
That was, it was just by, I wanted to do that before. I expected to go to Madagascar more than what I've been. And so,
Well,
thanks.
We have a question from online.
Yeah, oh, sorry.
Jordan was asked, all in use practices, after publishing new species, besides obscuring GPS coordinates and having to do that.
So, yeah, there was a great paper that was called "Do Not Publish," and that's the first step you take. So, for example, it was first collected in 2008, and we identified it as a new species basically immediately. We had a few back and forth on the genus because Solenangis was not present in Madagascar, and there was a few other genera that would the genus. We could describe the species because we had enough samples. Now the problem was like this species has the third longest spur in Madagascar, and this is the kind of thing that collectors like when it has something that takes it out of the ordinary. So we did not publish it immediately. We waited until we had enough plants in the shade houses and enough seeds in the seed bank before we published it. And it was indeed a good move because they're probably sold somewhere.
All right, well, Simon has a long afternoon in front of him, so we'll give him a break.